Hello, everyone. Welcome to Preserving Brain Health webinar. We're very excited. This webinar today is collaboration between McKnight Brain Research Foundation and American Federation for Aging Research. Just a few housekeeping rules, please. Attendee lines are muted, so if you have a questions, please put it in the Q&A box at the bottom of the screen. We are discussing research and opinions today, so we're not offering to serve any medical or pharmaceutical advice, and we will be recording this webinar so the next generations of scientists and entrepreneurs can revisit this again. We have two moderators today. I'm Sergey Young. I'm a AFAR board member. I'm longevity investor and book author, the book called The Science and Technology of Growing Young, and I'm very excited to introduce my colleague is Alice Luo Clayton, who is the Chief Executive Officer of McKnight Brain Research Foundation. Alice dedicated her whole life to the brain science. At some point of time, unfortunately, I couldn't go through all stages of her exciting career. At a certain point of time, she was a senior science advisor to NIH Brain Initiative, which is a $3 billion investment into understanding how the human brain works, which I think is incredible. So we have two hosts today for the webinar. I'm representing AFAR, American Federation for Aging Research. For almost 50 years, AFAR has been committed to progressing the science of healthy aging. AFAR is a field talent incubator. We provided more than $225 million to more than 4.5 thousand investigators, brilliant scientific minds to date. And this webinar is done in collaboration with McKnight Brain Research Foundation. It was founded in 1999. I love the mission of this foundation, which is preserving memory, enhancing life. For more than 25 years, the foundation has funded more than $200 million in research targeting age-related brain decline and memory loss. What is also interesting, foundation has this Innovator awards in cognitive aging and memory loss. And this grant program has supported 8 investigators in previous years. And again, it was done in partnership between AFAR and McKnight Brain Research Foundation. We are very lucky today because we have 2 out of 8 winners with us today. So this brings me to our 4 amazing panelists. When we were preparing for this webinar, I told them they all should write a book about their personal and scientific career. And I don't have time to tell all the stories about all 4 of our stars today, but I'll try. So Leah Acker, the winner of last year's Innovator Award. She's doing a lot of exciting work in Duke University School of Medicine. At some point of her life, she was a medical doctor. She was an engineer at MIT, and she's working on an important topic on how to protect human brain before, during, and after any surgery. Very exciting stuff. The next panelist is Denise Guy. She got the Innovator Award in 2023. She's with Icahn School of Medicine at Mount Sinai, focusing on how memories are stored inside our brain and what are the practical implications of this for all of us. At a certain point of her life, she was working on REM sleep and connection between REM sleep and creative problem solving, which is exciting. She's also co-developer of Miniscope, which is now in use by hundreds of labs around the world. The next is Amit Atkin. He's the president and CEO of Alta Neuroscience. That's a public company. He has a very special and important background for our field. He's a trained psychiatrist, And Amit dedicated many, many years of his life to build a platform which connects psychiatric treatments with personalized brain biology of every patient. And the next is Miriam Khairuddinova, CEO and the founder of Brainify AI. The mission of Brainify AI is to build the world's most comprehensive EEG intelligence platform. She's also a recent book author. Book was published, I think, this year, Miriam. And it's called Decoding Despair, How AI is Reshaping Psychiatry. Exciting panel, and I'll hand over the microphone to Alice to go ahead with the questions.
Thank you so much, Sergey. It's a real privilege to be able to be on this panel with these incredible speakers. And with you, I just want to say thank you to you and AFAR and our audience today for taking the time to be with us on World Brain Day. I think the topic of aging and longevity and healthy brain couldn't be more topical and timely right now. And so much has happened, even in just the last 5 years. And so I would pose as the first question to all of you, given your different perspectives, what do you think is the biggest single shift in the brain health and aging field in the last 5 years? And let me start with our basic scientist, with Denise, if you wouldn't mind.
Yeah. Well, thank you so much for having me. And first, I wanted to say I'm so grateful for the support from AFAR and McKnight that allows us to ask these questions. I think within basic biology, we're no longer just getting snapshots of the brain and behavior, and we're able to record, the same cells of the brain across the animal's lifetime. And what that means is we can make predictions of what is going to lead to, emergence of age-related deficits. And so the ability to be able to track across the lifetime will allow us to pinpoint the precise point when things go wrong. So that's what I'm really excited about.
Fantastic. Thank you. I'll turn it to Leah, who wears both a basic scientist hat and an anesthesiologist. So I would love to hear from you as well. What is your perspective on it?
Thanks, Alice. And I'll echo what Denise said. I am so grateful for the support from this foundation and for just the general support for aging research because it's so important. One of the things that I've noticed as a clinician is that people expect to bounce back from illnesses now. Aging is not simply expected to be this like linear decline. We really have an expectation now that, hey, you should bounce back at any age. And I think that's a really exciting thing that we are breaking down this idea of, sort of maybe delaying aging and saying, no, we should be able to reverse it. We should be able to avoid many of these declines that come, particularly with physical aging.
I love that. I love that. And I think that's a great pivot over to our two entrepreneurs. I'll ask Emmett, how do you think about it in terms of, you're a psychiatrist, researcher, entrepreneur. Tell me how you feel like brain health and brain aging is changing over the last 5 years.
So, my when I did my MD PhD, I was a basic scientist working in learning and memory like a long, long time ago. And in the time that I've spent just in my own career going from basic to more translational science in work that we were doing academically, and then bringing that into the clinic in also neuroscience, you can see a shift in the desire to intervene. You can see a shift in bringing measurement tools to be able to, what we would call biomarkers, to be able to characterize people. Now, part of what we and others are developing is a precision psychiatry approach, the idea being measuring something about a person that informs the treatment. And you also see a lot of novel mechanisms coming into play in humans. And so that shift from like, we've got to spend a lot of time characterizing things in order to understand them, to we understand enough to start making inroads. That's actually really exciting. And it's actually just beginning, which is perhaps even more exciting and the shift having been made.
Thank you. Yes, no, I think that's terrific. And then to Miriam, she's coming from a quantitative math background. So thinking about, the action piece of it and the quantitative sort of grounding that can drive it forward. Miriam, please.
I really like how longevity right now is shifting the relation of the patient to their brain health. Like literally a couple of years ago, people check their brain only in the case there's something significant happened. They have epilepsy or seizures or some significant brain trauma. And that was the only piece of data that we could get back then. But now with the growth of longevity and amazing clinics in longevity that we have in right now, I see patients being curious just to check their brain regularly to see that there is nothing abnormal is going on. And it's not only good that it can help them to improve their lifestyle, to see how their brain operates better, or check an abnormality activity early, but it's also good for the whole science field because it generates data. Generates data from relatively healthy people, something that we've been missing for a long time. And it gives us an opportunity to better understand how brain actually develops over the time, how disorders are progressing, and helps us to learn more how we can catch it earlier, diagnose it earlier, and treat it better.
Absolutely. Thank you. I'd like to turn now actually to some specific questions to our panelists. Each of you have such a incredible professional interests and expertise. Leah, as a anesthesiologist and cognitive neuroscientist, your Innovator Award is focusing on this concept that you've coined, inflammatory hangover. Can you kind of unpack that for us and talk about how you think it contributes to cognitive aging? And how you think how surgery became a window into better understanding the underlying mechanisms of cognitive aging.
Absolutely. Thanks, Alice. So one of the things that's really frustrating to me as an anesthesiologist is that I'll have two patients going in for very similar surgeries who look very much the same on the surface when I do the pre-op evaluation. And in the operating room, the surgery appears to go about the same. And then I would come and visit these patients the next day, and one of them would be exactly the same, and one of them would have a major cognitive deficit. And this kind of thing that maybe we were doing some harm unknowingly to some of the patients really ate away at me because that's the whole thing you're supposed to do no harm as a physician. And so I really dug into what is the difference between these patients. And Sir William Osler said, if you listen to the patient, they'll always tell you the diagnosis. And so I really tried to listen to patients and their families when they weren't really coherent enough to give me information to understand what was going on. And I dig into their lab work. And we found in a large study last year that some patients resolve inflammation after surgery quite quickly. And that's appropriate. You need some inflammation in order to start healing. But there are other patients, particularly older adults, who continue that inflammation well beyond the point needed to initiate healing. And that's what we call this inflammatory hangover. And so we and others have shown that there is a link between excessive inflammation after surgery and delirium and ultimately long-term cognitive decline. But most people don't undergo really big surgeries. They undergo a lot of little bumps in the road. Things like a small infection or a UTI or an upper respiratory infection, or maybe a small knee surgery. And so we started thinking, well, maybe there are people out there who their families would tell us, they were doing fine, but then just like bump, 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 and they went downhill. And so maybe there's inflammation that lags after each of those little insults so that in series, they have a cumulative insult more in line with what we see with a big kind of surgery or big illness. And so that's the whole premise of this inflammatory hangover. Really trying to get at why, when you talk to families, they'll say, oh, my family member was fine, but then just thing after thing after thing after thing. And they went downhill fast. And this isn't a population that NIH studies a lot. We don't study healthy people as much as we should. And I think that's a real problem because what do we ultimately all want to be and want our family members and loved ones to be? Healthy, older people. And so that's how we got there with the inflammatory hangover.
Absolutely fascinating. I mean, I think exactly. I mean, I think it's speaking to the broader audience of it's inclusive of all of us will get ill, but it's, it's part of life and you're going to, how do these perturbations impact us? So I guess I would turn it over to Denise to ask, you use animal models, and so it gives you an opportunity, a window in a very different way from how Leah does it, to understand how experience over the lifespan really changes the organism. Can you tell me a little bit about your work in animal models and using cutting edge technology to understand memory over the lifetime and how that has provided insights into cognitive aging?
Yeah, thanks so much, Alice. Also, yeah, that's super cool. And I think relatedly, the questions that keep me up at night are like how all these different experiences build up into whatever state we're in. Okay, so the question my lab is really interested in is how is our memory stable and flexible? And there's like this tug of war between the two. And there's a long running debate in our field about what memory really is. So one view is that the memory is static, it is permanent, it does not change. So you can imagine a memory is like a book that you slide onto the shelf and it sits untouched until you want to retrieve it. But my view or our work and others support that memory is actually quite dynamic. So let's say you have this book, well, you gotta maybe shuffle the other books to fit it in, and that reorganizes how things were from the past. And that reorganization is really important. We think of that as a healthy sign of memory, as opposed to memories being unstable. So the way we study this is using these like tiny little microscopes that we can put on an animal's head, and we can peer into the brain as they're interacting with the world, learning, and we can see hundreds to thousands of cells firing at the same time. And so we can capture the neural code of these memories and these experiences, and we can see how that relates to behavior. Okay, so since this is a aging seminar, let me give you an example of what we're looking at. And this is a real life example. So I used to live in LA. I used to go to Disneyland all the time. How many of you guys been to Disneyland? A lot of you guys. Okay, so you know that, right, there's the parking structure is huge. Luckily, Disneyland helps you because every level is a different Disney character. So let's say one day you go and you park Goofy row 2, right? Now, if you have a very precise memory, you remember, okay, I parked on Goofy row 2. If your memory is not quite precise, you're like, I think it's Goofy. Now you're like walking all over the place looking for your car. I've had that experience. So that is memory precision. And that's one of the first things to go wrong with aging. This happens even in middle aging, like around, humans like 60s, but in rodents, about 12 months. And that happens before general cognitive decline. Okay, but here is the flexibility, which is really important. And we find that this goes wrong in aging. So let's say you go back to Disneyland again, because it's so fun. And this time you park at level Daisy, right? Now, when you go back, your brain needs to forget that it was Goofy and that it's Daisy today. And the aging brain has a really hard time doing that. And that these two memories are blurring together. And this is what caused some of the flexibility or the inability to update memories. And so as we're getting older, the memory precision is one of the first things to go wrong. And so what my lab is really trying to characterize is one, Emmett talked about biomarkers, and we're trying to come up with behavioral measures when you can't go into the brain of humans, right, to make predictions about how are things going to go awry. The other things that we can record the brain before the animal ever does anything, while they're sleeping, while they're doing other things. And with that activity, we can actually make predictions of, will they remember well? Will they learn well? And so that's kind of, the body of our work, trying to understand this stability and flexibility and how this changes with aging. I'll just say one last thing, which I want to echo Leah, which is that this idea of preserving memory function and intervening early on, I think is really important. I think a lot of the treatments, especially in Alzheimer's, is too late. So much of the brain has gone wrong. And so if we can think about healthy aging as opposed to fixing a disease, I think that's something really exciting in the last 5 years.
Fantastic. Thank you so much. Just both of you are doing just fascinating work, and I love the themes of plasticity and resilience and the ability for the organism to respond in adaptive or maladaptive ways. I'll turn it to Amit. You're coming from, you're a psychiatrist, a neuroscientist, and an entrepreneur. Tell me a little bit, going back to Leah's comment about two individuals that can look very similar on the surface, but biologically, and their response to treatment, their response to life's bumps in the road. For those of us in the field, we talk about how we want to match the treatment and the patients together in what we call precision psychiatry. Can you tell me a little bit more about what you are learning through Alto Neuroscience about sort of both the commonalities and the distinctive aspects in precision psychiatry and how it could glean insights into cognitive aging?
Yeah, so I'll take you back a little bit to around 2010, which is when I started on faculty at Stanford. And at the time, we were looking at diseases as related to healthy people as one disease versus healthy people. No relationship of disease to disease, and really assuming that the whole category is defined symptomatically is a valid way to categorize people. What we started finding then is that actually diseases are more similar to each other than they're different in the brain, no matter how you measure brain function, brain imaging, and various other things. So then that turned the attention to saying, well, if all of these diseases that have different symptoms are similar in many regards in the brain, what about individual people within a disease? And they can have a wide range of symptoms. It turns out most of the way people express their symptoms is really as a idiosyncratic measure of distress. There's almost no mapping of individual symptom to brain function in the way that you would think. So the trivial example would be your sleep, right? When you're asleep, you don't have access to, what your REM is like that night. What you have access to when you report on it in the morning is how you feel about your sleep. So that's really more a measure of mood than it is of sleep. Turns out appetite, even though you know what you're doing day to day, cognition, even though you know what you're doing day to day, map very little onto objective measures of those capacity. So here, this is framed within the context of depression, but this could be framed in any disease context. And what we started finding is that if you say, all right, that's one way to characterize a person, there's their symptoms. Another way to characterize a person is their brain, and you focus on things that are stable about their brain. Cognition, for example, was measured, and that's one of our favorite ways to characterize people. Brain activity using EEG, brainwave recordings, which are a very scalable, low cost, stable way to characterize brain activity, all can characterize a person, what we would call a biomarker. Biomarker really just means measuring biology in a meaningful way. A lot of jargon around this, but really is trying to say, like, can we find the same person based on their brain profile, even when they may have very different symptoms, or the people with the same symptoms that have very different brain profiles, that starts to anchor our ability to develop drugs. So I'll give an example of one of our programs that actually ties together some of what you've heard around neuroplasticity. So we've been studying as a field, and as individuals here in this panel, the process of neuroplasticity. One of our drugs, Alto 100, was identified based on what's called a functional assay. So looking at its effects in a dish of neurons to be able to grow new neurons and connections between neurons. Turns out to do this pretty well. And you can see in animals a variety of pro-neuroplasticity effects. It affects signaling by growth factors like BDNF, which is a brain-derived neurotrophic factor, a very important growth factor in the brain. And when you give it to people, you see that some people benefit in terms of their mood and others don't. And the thing that actually predicts whether you benefit or not is that you have, on the one hand, in some people with depression, a deficit in aspects of cognition that relate to plasticity, in particular learning and memory. And in others, there isn't that deficit. They have the same symptoms, but one has a deficit in plasticity, one does not. It turns out the people with a deficit in plasticity seem to be the ones that respond best in terms of the mood effects of this drug, because we know that plasticity is important for both cognition and mood in the same brain structures. And so that already shows you how, with a very simple test of memory, the kind that we've been doing for a century, you can start to tell biological differences between people that could direct their treatment. In this case, we're developing it for bipolar depression, where all these people have available to them right now are antipsychotic medications, which carry a huge side effect burden. So the need to understand basic neuroscience, translate that to humans, and overlay on that the ability to guide novel treatments. This is an entirely novel mechanism of action drug. Makes this, I think, a very exciting time. And you can go from depression to any number of other disorders, now understanding the biology of the drug and the biology of the people and how that goes together, which seems awfully novel for our field. But actually, in areas like oncology, is kind of de rigueur. And if you go in and say, we do biomarkers in an oncology audience, they'll say, yeah, we just call that knowing what you're doing. And we're just new to that frame of reference, I think, within brain science as a whole.
Absolutely. No, I think that's exactly right. I think the mindset is changing so much, and the bringing together of the behavior and biology in these exciting ways is really sort of the next frontier. And I think all of you use technologies in such innovative ways. I want to ask, Miriam, I mean, you're a mathematician by training, and you have a company, Brainify AI. You're using AI and found the development of a foundation models, using it with these brainwave recordings to tell us a little bit more about the biology and to empower folks to take the health back into their own hands. Can you tell us more about your company and your approach?
Yeah, sure. You're absolutely right. We're mostly focused on building the foundational model for EEG, but most of the people don't understand what are the foundational models are and what is the whole idea of it. But the main goal for foundational model is to make the diagnostics and biomarkers scalable and generalizable. And let me explain, what does that mean? Like, if you look back for decades, most of the brain studies has been done on a very small and homogenic data sets, usually 100 or 200 people. And everybody mentioned already, brain is plastic, right? So there is no two similar brains in the world. And definitely the results, unfortunately, even promising, even looking exciting, they were not replicable, and they could not go from the academia and research labs into the clinics and hospitals. And that's the significant gap that we're having right now. And of course, when it even comes to more complicated disorders and complex processes, such as psychiatric conditions, for example, where is a lot related to perception, and where our brain is impacted by culture, by, different like environmental impact, etc., it is important to be sure that we know more about differences in the brain globally. And we know the whole diversity also on a scale of disorder progression, from the healthy controls up to the people who are like severely distressed, which means that we need some kind of basis that will allow us to understand the overall mechanism of the brain. So that's the idea of the foundational model. Foundational model is something where you're given the data not from hundreds or even thousands of patients. It should see millions and millions of different people with different types of the brain conditions. So usually, like, we're trying to achieve the global scale population. So we're trying to be sure that we cover in as many cultures as possible, as many countries as possible, that we have significant amount of the healthy controls, that we cover in the whole scope of the age differences in our data set. And then the foundational model, its goal to understand the universal language of the brain and eliminate the noise and keep the signal that is pretty much stable. And it gives an opportunity for pharma companies, for diagnostic companies, using this basis as a foundation model to develop something that actually going to scale. So I'll give you an analogy in GPT, because most people understand what is GPT. GPT is a foundational model that learned the mechanism of our language, how we build and how we talk. But it was possible only because of all this data available online, because it's been trained on 20 billions of pages. And that's how it learned, okay, where's the noise? Where's your typos? Where's the different structures? What is the main like rules of us talking in language? Same with trying to build for the brain, right? We're trying to understand this universal language. And then if you have this foundational model, we license it out to pharma companies. And for example, when they're doing their clinical trials, trying to stratify patients, trying to find the biomarkers, they can use it as a ground. So the foundational model will tell, okay, we're eliminating all the noise. This is something like clinical trial site specific noise or like your patient population specific noise. But this signal is something that you can actually rely on and will help you to produce a biomarker that will equally work with the same accuracy, not only United States, but also, for example, in Kazakhstan or Japan. And this is usually the goal for most of the medical devices companies and the pharma companies, right? We would like to be sure that whatever we're producing is actually accurate and stable and can translate in the real world population and the real world clinics and can stably work and give like reliable results. That's what Brainify is working on, and this is what our foundational models are for.
Amazing. There's such rapid progress in this area. And speaking of progress, we have all four of you are, we have the gamut from academic science through to the marketplace. And thinking about that progression in the pipeline, particularly when things are so uncertain right now in the US and globally, really. I'd love to know, from your different perspectives, how do you see the biggest bottleneck to progress now? There seems to be so much promise. Maybe I'll start with Leah, if you have any thoughts on from your perspective.
I think there's incredible work being done in the science, and it's not just the basic science and the mechanisms, so that's exciting. It's also in the data science. So this is a really exciting time. And so if we as a society continue to invest in this, and I know it's uncertain times, but I think we will, because I think that it has a lot of value. The problem then isn't that we don't have enough discovery, so we can never have enough. It's getting those discoveries to the patient. And there's a lot of things that are difficult in getting it to the patient. First and foremost, the patient has to want it, which means that they have to believe that it is a material chance of benefiting their lives. This is why a lot of people don't necessarily want to know if they have a biomarker for a certain disease unless there's something you can do about that disease. And we see this in our own studies. And it's true in most clinical cognitive science research. When you look at patients, albeit under stress because they're awaiting surgery, many, many more, and we're talking many-fold more, say that they have perfectly normal cognition and their testing objectively does not agree with that. Now, it could be the stress of surgery, but that's not the case. Sorry, my lights in my office are very energy efficient. Secondly, the issue is even if the patients want something, somebody has to pay for it. And so you have to show value to insurers, to health systems, to some of our big government insurers like Medicare and Medicaid. So you have to show that value. And then you have to have it be simple enough to get into the clinical workflows. And so this is why this whole field of implementation science has evolved. And so over maybe the past decade, my thinking has really evolved that I don't just want to do phenomenal science, although we all want to do phenomenal science, and we've heard a lot about that. We also need it to be simple enough that it can actually affect the patient. And it needs to be beneficial enough that patients actually want it. And so I think those are the challenges that we're really going to have to address.
Yes, no, absolutely. I'm curious, not to put you on the spot, Emmett, coming from your background as a psychiatrist, what are your thoughts on Leah's comments?
So I think she hits at a lot of really key issues, right? A lot of research in the earliest stages is curiosity driven. I actually think that there's often too little exposure of the opportunity, just to the people doing the research, of the opportunities that kind of research has for translating to patients. That's a really important place to to intervene if you were kind of thinking about this from a systems perspective, to bring more of those solutions to patients and inform likewise in reverse how the basic science could be structured based on the reality in the clinic. I think we need to be trying more things in the clinic. That's often a very real bottleneck, is for a range of very obvious and structural reasons, like showing safety of a new drug, even just the time and cost to develop a new drug, the cost of developing through clinical trials a drug, and so forth. You can only test so many things. The more things that we could test, the faster we could learn. I think understanding where the problems that really drive cost are and orienting to those in a much more systematic way. There are areas that we don't really touch at all that are major drivers of disability and morbidity and mortality as far as brain functions go. And for various reasons, research has not really been well funded in those areas. And there are sort of sexy areas and less sexy areas. Doesn't necessarily reflect where cost actually is and where patients' lives are. I think another underlying factor that is a barrier is actually cultural, is the culture of sort of academic culture, entrepreneurial or industry cultures, the perception of a bit of a gulf between them, which in some cases is real, in some cases is not, that prevents the creating of large data resources and sharing of data. It's actually one of the things that has been quite frustrating to me historically, is just how much data are collected by the field, by individual scientists, that doesn't make it into a usable, harmonizable form that everybody could benefit from. Like that if you think of just the NIH, investment in data collection, which on the NIMH side is, 2 billion a year or whatever it is these days, or whatever it should be these days. Uh, None of that really translates to something that can be used in a very broad, generalizable way. There's not the discussions between investigators and between companies and academics and what kind of data should be collected. How do we make sure it's harmonizable? And how do we make sure it's shared so that there is a collective knowledge that increases? It works kind of by fits and starts. I think we can be a lot more systematic, and by being more systematic, we're going to see more diagnostics, more therapeutics coming in, and just a much more exciting pipeline that goes all the way to basic science.
Absolutely. Thank you. I'd love to give Miriam and Denise a chance to respond, because I think they both use and advocate for open science and depend or generate huge amounts of data. Either one of you, please.
Oh, I'm happy to take from here. Thank you, Amit, for opening the stage with this problem. Yes, for me, the biggest bottleneck is always the data. Brainify right now have data for 500,000 patients. So far, it's one of the biggest data sets, well-labeled data enriched with HR, AMR, but it was quite painful for us to reach out to different clinics, institutions, academia to collect whatever there. And then we had to start to generate our own data, because most of the data is mostly people with disorders. And we need also healthy controls, right? We need people on a different lifespan. So that's how we got enrolled in a lot of wellness centers, longevity, fitness, where right now we are collecting more brain data, but from the people who are do not have significant conditions. So we can actually understand the whole lifespan and the brain development and the disorders. But there is also other issues coming with the data, not only the fact that it's complicated to harmonize it. We just built our own platform. We learn how to harmonize data, how to combine it from different sources. So we built a whole product around it. But there is also the bottleneck, because you would like to have people from different countries and different populations. And here we're bumping into regulations, into the different privacy policies, and the ability to build different infrastructures, which is crazy expensive. Because, for example, when I'm collecting data from Middle East, I must store data locally and I must process data locally. Same if I'm doing the data collection in Europe, I'm bumping into GDPR, because the brain activity, brain data considered to be personal data. So I'm not allowed to like transfer this data. I have to have a people who's going to be in Europe and do all the processes and the analysis over there. So all of those regulation things around the privacy and the data make it even more complicated, even when you have access to how to collect the data, but you still have to navigate a very expensive and complicated world of the infrastructure and the privacy policies.
So much.
Yeah.
And yeah,
I was just going to piggyback on, well, I agree with what everyone said. I think getting it to patients and this big data. So I have 2 points I want to piggyback off of Miriam. So one is, we're not lacking data. And now with our cutting edge tools in rodents, humans, just in my lab, I'm recording, let's say 1,000 cells, the activity every day for the lifetime of the animal across lots of animals. That's a lot of data. And so one of the questions is, how do you make sense of the data? And I think we really need more computational models and different ways of reducing the complexity of the data so that it can be meaningful. And I think what Miriam is doing is really amazing. The other thing Miriam talked about, and actually all of what everyone talked about, was, across scale, right? So one of the challenges I think with federal funding is that it's really hard to get funding to do human work and rodent work in the same grant. And unless we're talking to each other and working together, then it creates this barrier between what I do as a basic scientist and the clinical side. And I think more opportunities for people to be able to collaborate together or even share data and build the same language, I think is really important. Along that end, I think Miriam talked about global populations, right? And so you had mentioned that part of what we do is try to make our tools open access, but not just open access, but make it affordable and easy to use. And so we have several hundred labs around the world using our tiny miniature microscope. And I remember, so one of the things that we do is we travel around to the countries and we teach people how to build it themselves. And I remember I was in New Zealand. This experience actually transformed the way I think about open source tools. And there was a scientist there that said, Denise, in the US, it's only $150,000 per microscope. So we teach people how to make it for $1,000, right? So it's 1/100th of the cost. He said, but in New Zealand, the taxes requires to pay $250,000. And so how many labs have that kind of money, right? And so I think that made me realize in order for us to make discoveries that are generalizable across not species, but across cultures, we need people with different perspectives from different cultures, different questions, all have access to be able to ask and answer those questions. And so, I think access and communication across scales and across cultures is really challenging. And I wish there was more funding for it.
Always, always. I love ending on a really positive sort of call to action. I'm seeing all the great questions populating in the Q&A box. Please keep them coming. But I think at this point is a great time for me to hand it over to Sergey to moderate.
Thank you, Alice. This is very exciting. We have another 15 or 20 minutes, so let's just try to be short. To the extent you can be short to answering my next question. But I think it's a very important one and it's very exciting. Well, here's the question that we receive. Cognitive decline disproportionately affects women. 2/3 of Alzheimer's cases are female. What is the latest progress in understanding and addressing this disparity?
Yeah,
You want to go first?
Yeah, can I start?
Miriam, of course.
Yeah, one of the first studies that we've done, we were learning how to predict sex from the brain EEG. It was a very experimental thing to learn if there actually EEG has some information that is not seen by the naked eye. But it led us to understand how different is brain mechanism because it was so easy to understand who is it, male or female. And then we learned how much, also did the research before on how estrogen impacts the prefrontal cortex in the fear extinction processes. So I was kind of always in this women mental health life. So right now, I think we have challenges not about the even understanding the differences, but into translating them into the clinical trials. Meaning that I still see that the clinical trials are running 50-50 men and women, which is a little weird for me because I think there should be like a very distinct stratification that we need to develop drugs, including the sex and understanding the sex differences, and give them enough, I don't know, value, because some people think, okay, the disorder is similar among the sexes, which is not really true. And that's why the shift right now starts not into the understanding, but into the implementation, this in the industry level. So we will start treating men and women differently.
Can I piggyback on that? So, there's a call to be able to study, or there's a call to study both sexes, but a lot of labs still don't because it's very complicated.
Yeah.
And oftentimes what you find in the lab is that the behavior between female and male, let's say rodents, are very similar, right? They perform to equal levels. But when you look at the brain, how they solve that problem is quite different. And so, but it makes it complicated. And so, it requires, I think, more funding to be able to actually study both populations and consider them separately. But yeah, I think how the brain gets here differently across sexes, I think it's a really important question.
Amazing. Thank you. Yeah, Leah, please.
All right, I will jump in on this at sort of a very simplistic, like physician level. So women still consistently, or at least epidemiologically, live longer than men do. And so some of this is a tincture of time issue. Another major part of it, and I don't think we think about this nearly enough, is that the brain is part of the body. And most cases of dementia do have a vascular component. And we are much better at cardiovascular health and monitoring and treatment in men than in women. And so that's a gap. I'll be a little bit more hopeful, though, is that the female brain goes through a lot more over the course of a lifetime in terms of changes than the male brain does. There's been new work kind of looking at brain changes with pregnancy, which some have suggested can be a window into later brain changes because it's a time of stress. The same with menopause. And I think as we get more research into these brain changes at earlier life phases in women, we will start to get more insights that help us to alleviate the cognitive aging later in life in our female patients.
Oh, this is amazing. Thank you. We have a lot of great questions, so I'll just ask them in buckets. So the first bucket is about data, and this will go to Amit and Miriam. EEG has been in existence for the last 100 years, if I get it right from the internet. So what has changed in the last few years in terms of our ability to use this data and draw important conclusions? And there's the other, I think, related question from Warren is like, what is the definition of data today? Are we talking about dynamic imaging, biomarkers, behavioral, neural tissue data, etc.? So please, Amit and Miriam.
Yeah, so, it's an interesting thing when you ask that question, because I do get that question quite a bit of, what's changed with the EEG. Partly there is the computational that has changed, our ability to analyze complex signals. But actually, a more important part of it is the consistent collection of data in the right context. So when you look at how drugs are developed traditionally in, neuroscience and psychiatry, you maybe collect EEG data in your phase 1 studies, which is really looking in a small number of healthy people to see what are the effects of the drug on the brain. And mostly you're doing it for safety and to understand how much gets into the body and what the temporal profile of that is. It's been rare to collect data in a phase 2 study, which is really the first point where you see efficacy. And it's vanishingly rare, perhaps never been done before, to collect it in a phase 3 data set, which is really the kind of data set that you support approval with. And so the ability to get enough of the same type of data at enough in terms of stages of drug development, and also to contextualize that in enough data in the context of standard of care treatments, where we've not had much data following a patient over the course of even an acute antidepressant intervention, like data sets in the hundreds as opposed to the thousands, tens of thousands or more that you would really want. That's actually been a lot of what's held any type of data back, including EEG. And so it's really less about the technology and more about the context of use, the systematic thinking of how do I want to use this data? And let me make sure I'm collecting it in the right way. We try to collect data of all sorts, cognitive data, wearable data, genetic data, EEG data, blood samples. In every study, no matter whether that biomarker involves a particular modality or not, because it's building these data resources that you then are surprised by what it gives. Just one quick example, we have developed and validated an ability to predict who will respond to placebo, which is really important in a clinical trial, right? That comes out of the doing of this at scale across multiple different programs, and was not something that we ever designed for. So I think it's a lot of it comes down to mindset actually, and that is perhaps the easiest thing to change. At the same time, it turns out to be one of the biggest barriers.
Perfect. Thank you.
Yeah, I can't agree more. So regarding the EEG, it has been used for decades, but the way how we used it is very different. So we're here talking about the process and the computation, which now allowed us to see way more interesting information that has not been available and haven't been seen by the naked eye. And there is also like the amount of quantity and the quality of the data that we're collecting. As Amit mentioned, there are more well-labeled data, which is crucial when it comes to any machine learning training. And it's also about the quantity, which is coverage, generalizability, and scalability. So that's what changed. And like ability also to handle the noise, to start understanding where's the ground truth in the signal. That's what moving the field forward. And the EEG being relatively cheap and relatively scaled solution allows us to get to this amount of the data and that quality of data that moves this machine learning or data science possibilities forward as well. Regarding the second question, why we mean by the data, well, I categorize data in the three different fields. So it's biological data, everything that measurable and comes from our biology. That's what we mentioned by the data. It's kind of like molecular data, DNA data, whatever. It's definitely behavioral data, is the way how people behave. So it also can be used in the medicine. For example, their activity in the social media, the way how they like buy things on Amazon. It's still useful to predict the changes in their behavioral or their mood states. And the third type of data that also can be called as data is communication data. So the way how we talk, the way how our facial expression changes, our audio recording. You probably hear there's a lot of other solutions that using this kind of information, the way how we write, we text, talk, and our facial expression to predict or to see the significant changes in the patient states.
Thank you. Amazing. Yeah, Leah, please go ahead.
Just piggyback to give something a little bit more concrete for the audience. So I did maybe a dozen cases yesterday. One of them had intraoperative MRI, right? All but one had intraoperative EEG. And so if you just think about the prevalence and the ease of collecting this in a clinical setting, EEG is just really easy to get, and it's something we do routinely. So I think that is part of that answer.
Amazing. That brings us to the other bucket, which is basically around tools and tips. So if we can start with Denise and Leah, I mean, it can be broad. Is there anything we can do today that you would suggest to consider in terms of fighting age-related memory loss or brain function decline? There were a few specific questions, I think, from Charles on deliberate heat exposure. I mean, is it positive or negative in terms of the influence? So let's just go a little bit more practical with this question.
Yeah, so I don't know about heat exposure. I think it can be both. But I think the data suggests that, there are things that maybe are all very intuitive, but hard for us to put into practice. Exercise. Exercise, right, helps neurogenesis, makes regrow cells in the brain, decreases stress. Isolation is a huge stressor, social isolation. So to be part of a community and make friends. And the other thing is sleep. You know, Ahmed already talked about sleep, and we need to sleep to have our brains restore, also consolidate memories. And yet it's so hard for us to sleep. And I think one of the things like practicals, so I'm actually wearing an Aura ring right now, and I'm really excited about wearables, right? So in the lab, we can track the brains of, you know, all these animals across their lifetime. And as Leah mentioned, you know, how do you get treatments that are successful? Well, they have to want it and they have to have access to it. And I think for the aging population, it's really hard to always get them into the clinic to do the testing. And but if they can wear something at home and maybe get, you know, cruder results, EEGs or Oura rings or whatnot, I think that will be really helpful. It also gives them feedback. It might tell them, you know, predict based upon temperature when they might get sick. And I think having access to that kind of data, like every morning I wake up, I'm like, oh, what was my sleep score? And then I try to beat my sleep score, you know? And so having that feedback to your own data through wearables, I think could be really useful.
Thank you.
I,
Yeah.
I would agree with these general sentiments, especially with exercise, with socialization. Um, Ultimately, though, so when I anesthetize people, a 30-year-old is going to do well. But by 40, 50, there's this divergence, okay? I anesthetize a lot of 70-year-olds who act like they're 30. Occasionally, I anesthetize a 50-year-old who acts like they're 90. And it's kind of given me this insight that there are a lot of things that we do that are very minor in life that really matter. And so I would say numbers one, two, and three are exercise. And you don't have to go run a marathon. You need to walk your dog around the neighborhood or take a loop around the block a couple of times with your next door neighbor. This isn't fancy. And the better things are the easier things. I would go back again to this idea of blood pressure, diabetes, cholesterol. Get those things in check early. Maintaining a healthy weight, but not being too thin or too frail, because we know that frailty is a huge issue. But this doesn't have to be a super complicated thing. It's just building little things into life. It can be parking further from the grocery store and just those everyday interactions. There's also, I think, some data to suggest that it's good to interact with people who you wouldn't normally interact with. That's why it can be so good for an older adult to go and volunteer at a school to help kids who are struggling with reading, things like that. So I would advocate for not overthinking it or feeling like you're failing at preventing aging, but finding a few little things that you can do every day to help.
Yeah, and also
Perfect.
definitely sleep. The sleep is number one thing that you should do for your brain health and aging, like sleep properly 8 hours a day as regularly as possible, going to bed at the same time. Yeah, and checking on the quality. And another thing I would add to the exercise and the sleep is meditation. It's actually kind of a cool thing and it works well.
Love it. Thank you. Amit, I don't know if you have any views on that.
I mean, you know, I think it's been covered pretty well.
Yeah. Okay.
Hopefully, before too long, there'll be things that you can actually use that will be FDA cleared to
Mm-hmm.
you know to be able to track. I think one of the challenges is there's a lot of stuff out there of unclear validation and you know unclear meaning, and that I think causes a lot of anxiety. I mean, to Denise's Aura ring. Wearables give you a lot of information, much of which is not all that, you know, intervenable, and knowing what you can change, what you can't change, and not like there's a sort of wearable anxiety that some people develop, and knowing how to balance that is really critical.
Perfect. Thank you. We're going to be wrapping up in the next 5 to 10 minutes. I'll have a question for each of you in the end, but I just one of the question I think came from Roger. I'm personally curious of, like for the whole course of the webinar, we never discuss a blood-brain barrier, and there's a perception that it has a lower priority in terms of research or innovation. Is it true?
I don't think so. I think
Mm-hmm.
that the blood-brain barrier is actually a really important area of research. But inflammation and other things that we are researching do break down the blood-brain barrier. So I think we have
Mm-hmm.
hit on it, but we haven't been as explicit maybe as we could have been in saying that yes, we care about the blood-brain barrier because we care about the things that affect it.
Mm-hmm.
Yeah,
Perfect. Thank
and
you.
there's a world of new um, pharmaceutical strategies to try to shuttle things through the blood-brain barrier, which have not gotten through, which I think also, if you think of it rather than damage, but the opportunity to understand the blood-brain barrier and take advantage of it, and the ability to get biological molecules ultimately, like antibodies, into the brain, which is critical. If you think of like the Alzheimer's treatments that are approved, they're all antibodies. That is a whole new level of innovation that's just really beginning. But if you look at the utility of those kinds of treatments in other parts of the body, which are more
Mhm.
accessible, they've shown huge promise. So, this is going to be an expanding area for, I mean, easily the next 5 or 10 years.
Perfect. Thank you. Yeah, I think we have the same question from Robert. Thank you. So, for the final stage, we always end up with given advice or thoughts for the younger generation of scientists and entrepreneurs, because that's the purpose of this webinar. So I'll ask the question to each of you with the idea that if there's something we can offer to them as a way forward. So if you had unlimited funding and only one direction, one directive, what is the single most important research or entrepreneurial investment in a world of brain aging that you would do right now?
Okay, I'll go first because I just wrote a grant on this, which is I think we need to, in basic science, model real world life complexities. And so, for example, as someone who studies memory, a lot of time the rodent goes into one box and that's like the entire experience. And so, what I think would be amazing is given all the new tools to be able to really track the animals in naturalistic conditions. I call it Disneyland for mice, and that you can record how they interact with each other and look at how the brain changes in more naturalistic settings. And then I think we can, to scale, like actually generalize some of these ideas to humans.
Thank you. Thanks, Denise. Who's next?
Yeah,
So
I think, oh, go ahead, Leah.
things. So I actually kind of don't like this question. I'm sorry. I think it's important for everybody to decide this on their own. So I sort of don't want to
Hmm?
say what I think is the most important. But what I do think is really important, and this is the advice I give all of my trainees, and when I go give a talk, I give it as well,
Mm-hmm.
is to think about the pebble in your shoe. And so in the clinical world, I say that you should keep an app on your phone and write down one day one thing that you didn't understand or that bothered you. And then after a month, look and find what your pebble is in your shoe and pursue it. And so I don't think there's one thing that all of us should study. I think
Mm-hmm.
everybody should study something different that they are passionate about, but that they see the impact of this question remaining unanswered. So whether it's in clinical care, whether it's in when they're talking with their grandparents, whether it's when you're speaking with, your neighbors or your friends, what is that thing that keeps coming up that we need to answer? And whatever that thing is for whoever asked this question, that's what they should study.
Thank you. Thanks, Leah. Amit?
Yeah, so I'd say, I guess sort of in a similar vein, it's less the content and more the risk that you take. So,
Mm-hmm.
every scientist will come to a point where they decide, do I career-wise follow what I was doing? Content-wise, do I follow what I was doing, or do I see an opportunity there that suggests a fork in the road? And that may be a fork scientifically, a fork, you know, moving from academia to industry, or whatever the case may be, becoming an entrepreneur. Take more risks because that's really how opportunities are realized. And we have been I mean, certainly within science and talking about people getting MDs and PhDs, a lot of years in school, and you're sort of put into this path where you think that just following the path is all you can do. And in reality, you are probably the best trained person doing your thing out there, right? You are actually at the top of the tree, even though you don't feel that way as a young investigator. and you have a huge number of skills. So if you see the world for its opportunities, which involves risk, absolutely, but pursue those opportunities deliberately, I think, huge progress could be made on any number of fronts.
Thank you. Mariam?
My suggestion would be go after something that frustrates you the most. Because I truly believe those like little madness or being upset with something is the best motivation to change. Because like this positive motivation, I would like to make a world a better place, it disappears sometimes, especially when everything goes south in the wrong direction. You're like, okay, I don't need it anymore. But if you're actually very much frustrated with something, this drives you forever. So if you have a problem that's like triggers you, this is the way to go, and this is something to fight. So for me, it always was like an ability to be treated the way I would like to. Lack of understanding of the brain, lack of the data. I came from data science. I came from the math background into academia and neuroscience. I was like, really? We're looking at the 30 rodents and we're making decision. That was something that made me like mad and very upset. That's why I'm running my company, and I would continue doing this. But I truly believe that this is something that triggers you, that you need to follow.
Can I add one more thing? I realize I didn't answer this part of the question. I think what I would say is don't try to be anyone else, just be yourself. Because, right, like there is already a Leah Acker. The world doesn't need another. And I'm never going to be as good as Leah Acker, but the world doesn't have another Denise Kai, right? And so what makes science amazing is that you bring unique perspectives, and you're the only one that can bring your perspective. And so I think rather than comparing yourself to other people, trying to be someone else, figure out who you are. And so just be the best version of yourself.
Amazing. Thank you very much. What an inspirational note to end this event today. Just want to finish with thanks to our co-host, McKnight Brain Research Foundation, and Alice. Thanks to our panelists. We will share recording of this webinar on our website, on social media. You can follow us on LinkedIn and Facebook. And the last is your donation, big or small, are important for us to continue to offer these free webinars and help to support aging and brain aging research. Thanks for today. Stay healthy and happy.